Hello, class of Biochemistry 530 from Grand Canyon University. My name is William Grant Hale Jr., and today I'm going to talk about receptor tyrosine kinases and cancer. Before we get started, I do want to let you know and caution you this is not a beginner class. You should have some prior knowledge on the ligands and different types of some knowledge about tyrosine kinases as well. For the introduction here on this, I want to make sure you recognize that today I'm going to present the developments of receptor tyrosine kinases as they relate to cancer. The 8 topics I'm going to cover are going to help develop how these two relate to one another. Tyrosine kinases, early discoveries, also epidermal growth factor receptors, receptor tyrosine kinase with signal transduction, receptor tyrosine kinase in vivo functions, epidermal growth factor receptor deregulation in human cancers, a new discovery in HER2 new receptor tyrosine kinase cancer therapies, and then the final is receptor tyrosine kinase anti-angiogenic therapy. Hopefully, after we've gone through all of these, you'll have a better understanding of this topic. Now, with tyrosine kinases in the early discovery. Early discovery of the growth factor research began in about 1952 and resulted in a protein nerve growth factor being purified using snake venom and also mouth salivary gland extracts. It took about 5 more years of research for them to come up with the epidermal growth factor, EGF. Was identified. It was found to stimulate the proliferation of epithelial cells. Then in 1975, continuing on with the history, the specific binding sites for EGF and fibroblasts were found. The concept of tyrosine phosphorylation gained further experimental support, where in the early 1980s, the epidermal growth factor receptors, EGFR, and insulin receptor, INSR, and the platelet-derived growth factor receptor, PDGFR, are protein tyrosine kinases that can be activated by their respective ligands. So continuing on with this, now let's talking about epidermal growth factor receptor primary structure. Molecular clonings led to the identification of the clonal DNA that encoded important physiological peptide hormones and growth factors such as insulin-like growth factors for like 2-IGF-2, NGF, PDGF, and transforming growth factor alpha. But in 1984, the collaborators isolated and characterized the clonal DNA sequence of human EFRGR, which is the prototype for the RTK from normal placental cells and A431 tumor cells. High-level similarities between the EGFR peptides and avian oncogene. It is the first time that the animal oncogene with the human gene encoded a cell growth controlling membrane protein. When the complete sequence was cloned and completed for EGFR cloned DNA, more information was obtained. Truncations and deletions and mutations, things we've already studied, in the beta oncogene were identified. EGFR clonal DNA provided the first complete sequence of a cell surface receptor with signal generating ability and some insight into molecular structures. Also, they had a southern blot analysis also provided the amplification of the human A431 epidermal carcinoma cells. In spite of their unique biological roles, RTKs are highly related in both structure and domain arrangement and are similar to the EGFRs. Now we come to the receptor tyrosine kinases, RTKs, signal transduction. The RTK molecular mechanism presented a significant problem for the researchers, and it was found that the EGFR kinase domain of the chimeric protein was activated by insulin binding, showing that each of these RTKs use close related mechanisms for signal transduction across the plasma membranes. So the bivalent ligand was found for the vascular endothelial growth factor VEGF receptor FTL1 and the NFG receptor TRKA. The EGF stimulated the RAS oncoprotein to become active. The SH2 domain bind with a tyrosine phosphorylated peptide in vitro. You see the little picture right there. Now we talk about the receptor tyrosine kinase, RTK's signal transduction is continued on. Then we talks about in the 1990s, several mammalian cytosolic factors catalyzed the exchange of the RES-bound GDP and the GTP. With more thorough studies on the RES, the RAF, and the MEK, insights into how these RTKs regulated extracellular signal related to kinases, NRKs. So then also the RTKs were also found to activate the P13K, the mediator survival AKT, and the signal transducer and activator of transcription. Heterologous signals were also stimulated by the EGFRs and other RTKs. EGFR signal transactivation pathways were found to be involved in also the hyperproliferated diseases, including these cancers. This is kind of what we're dealing with here. This established that the EGFR functions as a point of convergence for diverse signaling pathways with key biological outcomes. Continuing on with this, we had the with these you can see that picture there for a cancer cell. This is a representative of the cancer cells there. Okay. Oops. Okay, sorry about that. The epidermal growth factor receptor deregulation in human cancer. It says early on it was found that the EGFR was for overexpression in epithelial tumors, show that deregulated EGFR signaling had an important role in the human cancers. And then the increased stimulation of the EGFR through autocrine growth factor loops was a common mechanism of RTK deregulation as well. The most prevalent mutation was found to be the EGFR number 8, which resulted from the gene rearrangement of or alternative messenger RNA splicing. Impaired receptor down regulation was found to be another RTK deregulation mechanism, and then the oncogenic forms of the ligase CBL functions as dominant negative mutants that prevent it from negatively regulating RTKs. Now we had a new discovery, the HER2-neu. That complete primary structure of the putative shows high levels of homology to human EGFR related to the HER2s. The neu was an oncological significance, because the monoclonal antibodies, the ILM-ABS, reverted transforming effects in the NIH3T3 cells. There's also a correlation between the HER2 overexpression in tumors and also in the reduced patient survival and the time it takes for it to relapse, if it does relapse. EGFR activation induces HER2 through the heterodimerization. HER2 is the preferred partner for the EGFR. HER3, HER4 was also partnered. However, the HER2 provides an extra recruitment mechanism of diverse intercellular signaling pathways. Now we're talking about the receptor tyrosine kinase, the cancer therapies. The HER2 gene is in breast and ovarian cancers, provided opportunities to develop target-specific cancer therapies. The humanization of this ILM-AB4D5 with therapeutic antibody development was then possible. Success was found with several medications in breast cancer patients, proved to be that it was able to target human oncoproteins. Then the anti-HER2 monoclonal antibodies, which interfere with the oncogenic signal generation was developed. This breakthrough led to the colorectal cancer treatment development as well. Tyrosine receptor inhibitors were also developed to help small molecule anti-cancer treatments. Very important breakthrough. Continuing on with this, the mutations in the ATP binding pocket of the EGFR abrogate its tyrosine kinase functions and interferes with oncogenic signaling. This led to also developing a small molecule tyrosine kinase inhibitors as well. This proved to be the fourth of this class of compounds for selective inhibiting of these cancers, and these active non-receptor tyrosine kinase is expressed in chronic leukemia cells, CML. In addition to the ABL and the BCR-ABL, the RTKs, PDGFRs, and the KITs were found to be potentially inhibited by some medications as well. Now with the receptor tyrosine kinase, the anti-angiogenic therapy, the VEGF and its receptors have important functions in the regulation of tumor angiogenesis, proving that the FEGF and the FEGFR signaling are required for tumor angiogenesis. The dominant negative VEGFR2 mutants block the subcutaneous growth of these glioblastomas. This led to the clinical anti-angiogenic therapy targeting either the ligand or the corresponding receptor. The VEGF and the VEGF and the VEGFRs were then identified as important targets for the therapeutic intervention in these tumor growths. These small molecules antagonists such as the SU5416 competitively block these ATP binding to the tyrosine kinase domain of the receptor, inhibiting tumor angiogenesis in vivo. Now that takes us just about to the end. I'm going to go through a little bit of the conclusion. Now that we have covered this, we've covered the tyrosine kinases, the early discoveries, the epidermal growth factor receptor, EGFRs, receptor tyrosine kinase RTKs, signal transduction, the receptor tyrosine kinase in vivo functions, epidermal growth factor receptor, the deregulation of human cancer, a new discovery in the HER2/neu, the receptor tyrosine kinase cancer therapies, and the receptor tyrosine kinase anti-angiogenic therapies. I think you should now have at least build up a little bit of a more understanding of how these tyrosine kinases relate to cancer. I'll go over a little bit with you with my references here. You see the first three references are basically from some academic journals. We have a textbook is their fourth, the reference there. And then the last two are once again are from academic journals. Well, I hope everyone has enjoyed this lecture as much as I have. This completes the lecture for the receptor tyrosine kinases and cancer. Once again, my name is William G. Held Jr. Just remember, life is good and be nice. Thank you very much.